we're looking at various different models, including the impacts of exposure to cannabinoids during sensitive periods of brain development, like prenatal development, adolescent brain development. And we're also looking at modulatory systems of the cannabinoid system, including systems like the fatty acid binding protein system, as an example.
And so I want to give everyone a sense for how lipids, fats are used in the brain. As some listeners will know, but many may not know, the endocannabinoids themselves are an example of how fats are used in the brain for signaling rather than just structural purposes. But when we think about endogenous cannabinoids, when we think about some of the lipid signaling molecules and related things we'll talk about, can you just give people a sense for how the brain uses fats beyond just structural components of membranes and things like that?
Right. No, it's an important question. It's often overlooked because I think we tend to focus more on the neurochemicals and the neurotransmitters and sort of ignore the bigger picture. Fatty acids are really the major. I mean, the brain is mostly fat. It's mostly fat, obviously including myelination and a lot of fatty acid signaling molecules and whatnot. And beyond, of course, the structural importance of fatty acids, which make up most of the brain, the cellular membranes and whatnot. There's a lot of fatty acid signaling molecules in the brain as well. And of course, when they have to traverse through an aqueous substrate, like the intracellular milieu, for example, we need to have specialized proteins that can sort of chaperone those hydrophobic molecules across these aqueous membranes. So that's why we have a lot of different fatty acid signaling systems in the brain, including the endocannabinoid system that relies on these specialized proteins that allow the signals to get through to where they need to go in the brain.
Yeah, so everyone knows sort of oil and water don't mix well together. Fats and aqueous things, watery things, don't mix well together on their own, and that's why I guess another way to get into this and think about it is the same reason you sort of have these chaperone lipoproteins in your blood that shuttle cholesterols and fats around. We've also got various things in the brain that help us shuttle lipids around. And one of the reasons we do that the nervous system has to do that is because these fats aren't just components of membranes. They are signaling molecules that actually help the brain process and transfer information around.
Absolutely. And of course, they also serve as important precursors for a lot of different neurotransmitters. So they're involved in the biosynthetic pathways of a wide variety of neurotransmitters, including the naturally occurring cannabinoids in the brain.
And so what exactly can you just give everyone a crash course in what the cannabinoids are at the molecular level and where they ultimately come from?
Right. So the endocannabinoids are, they're one of the most evolutionarily oldest systems in the vertebrate brain. And they're seen, across the entire evolutionary spectrum. And what they primarily do is regulate signaling in the brain. So they're primarily sort of retrograde modulators of neurotransmission. So, for example, the brain has to maintain a balance between excitation and inhibition. So the endocannabinoid system is very importantly involved in regulating inhibitory processes in the brain. So the presynaptic neuron will release endocannabinoids. And the two major endocannabinoids are called 2-arachidonoglycerol, or 2-AG, and then anandamide is the other major one. So those are the two major sort of naturally occurring endocannabinoids. They serve slightly different functions. We're still trying to figure out exactly what they do. But we know that both of them are involved in sort of this retrograde inhibitory modulation of the synapse itself, and primarily involved in sort of regulating excitation and inhibition balance in various brain areas. And of course, they're highly regulated in areas of the brain that are responsible for emotional processing, for cognitive processing, like the frontal cortex, the amygdala, for example, the striatum. So you can imagine that disturbances in the balances between those endocannabinoids are major risk factors for a lot of different mental health disorders.
Yeah. And I guess bird's eye view, the brain, there's information flowing through the brain in the form of, electrochemical activity. That information needs to be patterned. It needs to be structured and constrained. And in simplified terms, you don't want overexcitation. You don't want underexcitation. And the cannabinoids are an important mechanism preventing things from getting out of hand or underhand.
Exactly. Yeah.
And so, like, how does our body make them? Like, what are the pieces that go into producing them, and where do they ultimately come from?
Well, two of the major precursors for the endocannabinoids are DHA and EPA. I won't attempt to pronounce the full name, but those are, of course, the omega-3 fatty acids. And the omega-3 fatty acids primarily come from the diet. So, I'm sure you're aware that there's a lot of people are deficient in omega-3 fatty acids. They don't get enough in their diets. And of course, we rely on those omega-3s to sort of serve as the EPA and DHA to serve as the precursor molecules for some of these naturally occurring endocannabinoids. So it's a very complex biosynthetic chain. But ultimately, we do need to have those dietary precursors to get the adequate production of the endocannabinoids.
I see. So it's the omega threes, not the omega sixes, that ultimately are the precursors to endocannabinoids.
The omega sixes also are involved in some of that pathway as well, but it's primarily believed to be the omega threes that are sort of the major, especially DHA and EPA, which serve as the major precursors.
I see. And so when these things are used in the brain, so endogenous cannabinoids are famously made on demand. So as opposed to a lot of other chemical messengers in the brain, like neurotransmitters, they tend not to be packaged and sitting around waiting to be used. They tend to be made on demand. So are they basically just kind of clipped off of fatty components of the membrane?
Pretty much, yeah. I mean, it's a complex pathway, but they're primarily, you're right.